Hey guys, thank you for joining me in another IGCSE biology revision video. Today I just want to go through a couple of MCQ questions regarding the topic of plant nutrition. So what I'll do is I'll flick through the questions quickly, and as I do so I want you to pause the video, arrive at your answer, and I'll go through the answers individually. So here we've got the first question, the second question, the third, the fourth, the fifth, and lastly the sixth question. I can enlarge that for you as well. So hopefully you have arrived at your answer. So let's just go through this first question, which is to do with the limiting factors of photosynthesis, and the answer here was C. The reason why the answer was C is because when you get any of these graphs, initially you'll find that the graph sort of rises and elevates, and so at least in the first regions of these graphs here, you can see that the rate of photosynthesis increases with light intensity. That suggests that the rate of photosynthesis is dependent or reliant on light intensity. But at some stage it sort of plateaus out, or the graph sort of flattens, which the point P is placed around that region. And when the graph flattens, that suggests that even though light intensity is increasing, it's not actually affecting the rate of photosynthesis. That means that something else is now sort of limiting the rate of photosynthesis, and light intensity has no bearing on that. So whenever you get this sort of graph here, the initial stages will always suggest that that particular variable is the variable or the limiting factor of rate of photosynthesis, whereas when it flattens, then something else must be the case. And here is that scenario. We've got P, which is on the flattened part of the graph, and that suggests that it's not light intensity, it's the other factors such as temperature and carbon dioxide concentration. So the answer here was C. This question here is the second question regarding your knowledge about photosynthesis and and respiration. Respiration obviously produces carbon dioxide as a byproduct and uses up oxygen, whereas photosynthesis produces oxygen and uses up carbon dioxide. The answer here was C. The answer was C because the first two test tubes have no light that's getting through, so photosynthesis can't happen with these inside these water plants. So respiration is happening, at least in A, with the snail producing carbon dioxide and even the water plant producing carbon dioxide, but nothing is actually being absorbed back because photosynthesis is not happening. So therefore carbon dioxide is not used, and we'd expect it to elevate rather than decrease. So A and B would not have been correct. If you can consider the scenario D here, you've got a snail inside, which is obviously going to respire and produce carbon dioxide that it's going to dissolve into the liquid here, but there's no plant or anything like that, and the photosynthesis is obviously not going to happen here, and so therefore carbon dioxide is just going to start to increase as well. C, you've got this plant which is expected to respire, but it is also going to photosynthesize because it has access to light, and when that happens, carbon dioxide is still going to be moderately used up, and you could expect the rate of photosynthesis to be higher than the rate of respiration, simply because plants don't really need to respire a lot, at least compared to animals. And so therefore C would have been the most likely answer, with carbon dioxide being used up from the photosynthesis that's happening, and not much carbon dioxide being produced. This question here is a very, very basic question regarding your knowledge about the formula of photosynthesis, and here the answer was B. Hopefully there's not much explanation to do here. We've got the photosynthesis equation going, carbon dioxide and water being produced, or sort of basically producing glucose and oxygen in the procedure, and so obviously the answer would have been B. So we've got the diagram that shows a section through the leaf, and all you needed to do was sort of label what each of these things were, and so the answer here was B. And this really just tests your structural understanding of a leaf. And we've got a quick diagram here, just to recap. We've got the cuticle on the very top surface of the leaf, and then comes the upper epidermis. Below that you've got the palisade layer, which is of course compact, and it has a lot of chloroplast, and the main function of the palisade mesophyll layer is for photosynthesis. You've got the spongy mesophyll layer that has a lot of air spaces, and the main purpose here with the air spaces is for the diffusion and exchange of gases. and you've got the lower epidermis, and sometimes you've got another cuticle layer, and you've got guard cells that basically form the little hole called the stomata, where gases go in and out of the leaf. So if you take a look at this diagram here, we should consider the fact that we've got P being the cuticle, and so really the only answer here would have been B to start off with. So the only thing that could have confused people would be Q here, which is, you know, pointing towards some part of the vascular bundle. But if you think about the xylem being generally at the top part rather than the bottom half of the vascular bundle, then that would have given you the answer there as well. But overall, a pretty easy question. And so this question, fifth question here, the answer was A. And the answer was A because this really is, again, testing your knowledge about the leaf structure and sort of the fundamental functions of each of the layers of the leaf. And we touched on this right before with the diagram I showed you here. And so the palisade mesophyll layer is the main site of photosynthesis, so it's got the most chloroplasts, and hopefully you are aware of that. Now the second most layer that has the second most chloroplasts within the leaf is the spongy mesophyll, where photosynthesis can still happen. It's not the main function because it's got a lot of air spaces that is used for gas exchange mainly. So this question here is just really testing your knowledge about chloroplast distribution throughout the leaf. So without even looking at the questions or the answers here, we can see that 2 must be the palisade mesophyll, and 3 must be the spongy mesophyll, and 1 whatever is remaining. So that really just gives us the answer of A there. We've got the guard cell, we're having lowest amount of chloroplasts, we've got the palisade mesophyll having the most, and the spongy mesophyll having the second most. And this last question here, the answer here was C. And the reason it was C is because this question is really just testing your understanding of how to set up experiments in terms of rate of photosynthesis and all that. And one of the reasons, or one of the methods in measuring the rate of photosynthesis is measuring how much bubbles are formed per minute. Because the bubbles signify oxygen production, and we know that oxygen production is a result of photosynthesis. And so the investigation was repeated several times, and the average amount of gas collected was calculated, which 2 factors was kept constant during this investigation. So during any sort of investigation regarding rates of photosynthesis or respiration or any sort of cellular metabolism, we need to keep every other thing that can potentially affect the rate of that particular metabolism exactly the same throughout the experiment. And so here, if you think about the rate of photosynthesis, what makes most sense is that we keep the size of the aquatic plant constant, because if you have a larger mass of the plant, then you'd expect that would produce more oxygen, and the rate of photosynthesis may turn out to be higher because it has more cells in them. The other thing is the duration of light. Obviously, light is an important part of photosynthesis. You need light for that to happen. So again, if you put one in dim conditions and one in lit conditions, the one in lit conditions will mostly produce higher rates of photosynthesis, and that wouldn't really give you an accurate measurement or a test. So the variables that you need to keep constant are obviously the aquatic plant size and the duration of exposure of light. So the answer there was C. So I hope that helped you guys out. Please check out my Patreon if you want access to all these MCQ questions. I have a lot more if you subscribe to that. But otherwise, I will see you in the next video. Please like, share, and subscribe. Cheers.